Welcome to the Deep Dive, your shortcut to being truly well-informed. Packed with surprising facts and just enough humor, hopefully, to keep you hooked. Today, we're diving into, well, the revolutionary world of mRNA-based medicines. Now, if you've been following the cutting edge of biotech, you know the promise here is nothing short of incredible. We're talking about the ability to essentially program our bodies to create their own therapeutics, designing them to encode any protein we want. Think about that. Plus, a single strand of mRNA can be translated into many, many copies of that protein, so that means you often need a much lower dosage. And crucially, from a safety angle, mRNA can't integrate into your own genome. That addresses a major concern with other gene therapies. It sounds like a dream on paper, right? But in science, there's always a but, isn't there?
There absolutely is, yeah. And that's what makes the timeline so surprising. I mean, mRNA was discovered way back in 1961, but the very first FDA approval for an mRNA drug, not until 2020, nearly 60 years later. For the COVID-19 vaccines, that's a huge gap. So you have to ask why. What were the core scientific hurdles keeping this amazing tech in the lab for decades? It always came down to delivery. See, mRNA is incredibly fragile. It degrades super fast in the bloodstream, and it's got that negative charge, so it just can't cross cell membranes by itself. Plus, if you don't deliver it right, it can actually trigger unwanted, maybe even harmful immune responses.
Okay, right. So that's where the real breakthrough comes in, the kind of unsung hero that finally made these therapies possible. Lipid nanoparticles, LNPs. These tiny microscopic fat bubbles are the secret sauce. They protect the mRNA, shield it, guide it, and their safety and efficacy, proven globally. Moderna, Pfizer, BioNTech, COVID vaccines, so they both use them. It just shows sometimes the biggest leaps aren't just a new drug, but figuring out how to get it where it needs to go.
Exactly. And if we dive deeper into those LNPs, the ionizable lipid, or IL, that's really the most essential piece for mRNA delivery. It has this delicate balancing act to perform. It helps encapsulate the mRNA, obviously, but it also influences where the LNP goes in the body. It interacts with proteins, forms this protein corona, kind of like a biological zip code guiding it. But, and it's critical, it's also responsible for speeding up something called endosomal escape. Now, that might sound technical, but honestly, endosomal escape is one of the biggest barriers in delivering nucleic acids like mRNA. Think of it like getting a message into a super secure vault. You get it inside the building, sure, but if it gets stuck in the mailroom and never reaches the vault, useless. That was happening with mRNA. Studies showed tons of nanoparticles get into cells, but only a tiny fraction actually escape that internal mailroom, the endosome, before getting destroyed. If the mRNA gets trapped, it's game over.
Right, okay. So getting it in is only half the battle. The real trick is freeing it once inside, which naturally leads to the question, what if we could design new lipids that are better at this escape act? What's the cutting edge there? And that's exactly what we're diving into today. We're talking about branched endosomal disruptor, or BEND lipids, and their potential as a really significant step forward. Our mission today is to unpack how these new BEND lipids improve mRNA delivery, how they boost gene editing, and even revolutionize T-cell engineering. It feels like a major leap.
So yeah, let's just reiterate the main challenge. The mRNA LNP gets into the cell, gets wrapped in this bubble, the endosome. To work, the mRNA has to break free, get into the main part of the cell, the cytosol, to be translated into protein otherwise. Trapped, degraded, useless. Now, previous research on these ionizable lipids, it mostly focused on, tweaking the core amine structure, adjusting the length of the lipid tails, maybe their saturation, mostly the inner parts.
And this is where it gets really interesting. There was this existing idea, a hypothesis, that maybe lipid chains near the amine core, if they branched out, might help with escape. The thinking was they could form a sort of cone shape, disrupt the membrane, but then new findings suggested something else. Putting branched groups at the very ends of the lipid chains could seriously boost mRNA transfection, getting it taken up and working inside cells. The roadblock, though. They didn't have a good way to make lots of different types of these lipids easily. The existing methods were slow, expensive.
Exactly. And that's the core innovation from our source material here.
Sure.
Researchers developed what they called a facile, affordable, and modular synthetic approach. Basically an easier, cheaper way to build these lipids, allowing them to make lipids of various lengths with different kinds of branching right at the ends. And these new lipids, they named them branched endosomal disruptor BEND ionizable lipids. It really opened up the design possibilities.
Okay, so they have this new toolkit. How did they test these BEND lipids?
They made 12 different BEND ILs, right? Different branches, different lengths, and compared them to 4 standard linear ones, putting them all into LNPs, carrying a reporter mRNA, something that makes cells light up, basically, firefly luciferase, so they could see, Does the structure actually change how much protein gets made?
And the initial findings were quite telling. Despite all these structural changes in the lipids, they found no noticeable differences in the basic LNP properties. They checked everything: size, charge, uniformity, encapsulation efficiency, which was over 80% for all of them, size around 70, 160 nanometers. All looked pretty similar.
Interesting.
Important, this strongly suggests that any performance boost they saw later wasn't just some random effect of how the LNP formed, but was actually down to the branched lipid architecture itself.
Okay, that makes sense. So what did that mean for actual delivery in vivo in a living system? These numbers sound pretty impressive on paper. They were impressive. The results in mice were really quite something. With the first set of ILs, the 494 core ones, the Bend LNPs performed as well as, or even better than, their linear cousins. One in particular, E4I-494, actually had 1.5-fold greater total liver flux than C12-200, and C12-200 is considered a gold standard for liver delivery, so already an improvement. But then they used a different core, the 200 core, which is known to be more liver-tropic, and the results there were even more dramatic. All the Bend 200 core LNPs beat C12-200 for full body and liver flex. And get this, they even outperformed the lipid formulations used in the Moderna and Pfizer-BioNTech vaccines in these mouse models for liver delivery. One specific Bend lipid, E4S200, induced a 5-fold greater liver signal, just huge amounts of protein production. And those with isopropyl branches like E4I200 and E8200 were consistently good, beating C12-200 by about 4-fold.
Wow, okay, 5-fold is significant. Now, delivering mRNA for protein is one thing, but what about that next step, gene editing? Can these Bend lipids also boost the delivery of something more complex like the CRISPR-Cas9 machinery to actually edit genes?
That's exactly the next question they tackled. They used a mouse model for a condition called TTR-mediated amyloidosis. Basically, the liver overproduces this protein, TTR, which can build up and cause heart failure. Now, there are Serena therapies using LNPs to silence TTR, but they usually need repeat infusions. Gene editing offers the dream of a single-dose therapy. And the results were really compelling. The control LNP using C12-200 cut TTR levels by about 80%. Pretty good. But 4 of the Bend lipids cut TTR by an even better 80 to 90%. And they also caused more gene edits, 60 to 70% insertions or deletions or indels, compared to 55% for the control. And here's a fascinating detail. Remember how C12-200 is a gold standard? They also tested a linear lipid, C8-200, which usually performs very poorly for TTR reduction, like less than 10%. But when they added Bend structures to it, it became significantly effective. It just proves that these small molecular alterations can cause substantial changes in LNP efficacy, and crucially, toxicity. Two of the best Bend performers, E4I-200 and E8-200, both of those isopropyl branches, showed much lower liver toxicity markers, AST and ALT levels, than the C12-200 control. That combination of high efficacy and low toxicity suggests real translational potential. That's huge for moving towards clinical use.
Okay, this is incredibly promising. High efficacy, low in toxicity. But how? How do these Bend lipids actually pull this off? Is it how the LNPs form? How they travel in the body? What's the mechanism?
Right, the why. They did a lot of work on this. Rigorous mechanistic studies, and they ruled out several possibilities. First, no correlation between how well the LNP worked and its basic physical properties or shape, size, charge, whatever, didn't matter. Second, stability wasn't the key. They tested them in conditions mimicking the body, like PBS or serum, and the Bend LNPs were just as stable as the linear ones. Third, how they got to the liver was pretty similar too. Both Bend and linear LNPs use that ApoE mechanism, latching onto that protein to get into liver cells. And uptake by a liver macrophage or Kupffer cells wasn't a major difference either. Most importantly, the actual amount of LNP accumulating in the liver, roughly the same, whether it was a Bend lipid or a linear one.
Wait, so they get to the liver in the same amounts, their basic properties are similar, what's left? How do you explain the massive increase in mRNA actually working? This feels counterintuitive.
It does, doesn't it? But what's fascinating here is that the critical difference really boiled down to enhanced endosomal escape, getting out of that internal bubble. They showed this in a couple of clever ways. First, artificial endosomes. Using these lab-made mimics of the endosome, they found the Bend lipids cause 2 to 3 times more fluorescence signal compared to linear ones. More fluorescence means more disruption, more escape. Then they used computer modeling molecular dynamics simulations. These gave molecular-level insight into how it works. The simulation showed Bend lipids achieved substantial increases in bilayer penetration. They could dig into membranes better. You could actually see in the simulation snapshots. The Bend lipid tails could more fully extend into the bilayer, like little anchors digging in, whereas the linear lipid tails tend to just reside closer to the phospholipid surface, not penetrating as deeply. That's the direct evidence. They're better escape artists.
Better escape artists. I like that. Okay, so if that's the core mechanism, does this versatility extend beyond just liver mRNA? What about other tough cargo like proteins or other critical cell types? Could this Ben&D platform be a general solution?
It certainly looks that way. The platform shows remarkable versatility. They tested two other really important areas. First, delivering the CRISPR-Cas9 enzyme itself as a ribonucleoprotein complex, or RNP. Delivering proteins via LNPs has usually been harder than mRNA. But again, the Ben lipids helped. Specifically, E4I200, E4S200, and E800 enhanced LNP-mediated RNP gene editing in the liver. E4S200, for example, gave 1.5 times more liver signal than the C12-200 control. Interestingly, the control actually showed more editing in the lungs, but the Ben LNPs gave much better liver-to-lung editing ratios. So the Ben structure actually seemed to shift the tropism, making the RNP delivery more liver-specific, which is really useful. They also saw better cell-specific editing. E4S200 edited over 10% of the hepatocytes, twice as much as the control. And E8I200 hit 40% editing in certain epithelial cells, again, twice as much as the other LNPs. Then the second area, T cells, showing potential for non-liver-related applications, hugely important for immunotherapy. They tested BEND LNPs in human primary T cells. The BEND 494 core lipids performed as well as or better than the linear ones.
And crucially, with similar or lower toxicity, remember that linear lipid C8494 that was toxic? The BEND-E versions were much less toxic, but still boosted the signal. And one optimized BEND LNP, called E10I492B10, transfected over 80% of T cells. That's a fantastic number for primary T cells.
Over 80%, wow. And does the escape mechanism hold up in T cells too?
It does. They checked using confocal microscopy that really affected BEND LNP E10I494B10. It resulted in about 20% less of the mRNA being stuck with endosomes compared to the linear control. So less colocalization means more escape. It strongly supports that increased endosomal escape is likely a dominant factor for the BEND lipids success in T cells too. It seems to be the core advantage across different cargos and cell types.
Okay, so wrapping this up, this deep dive really shows that BEND lipids are a significant step forward. A new design principle, really, backed by solid mechanistic work. We've seen they can boost delivery for mRNA, for protein cargo like Cas9 RNP, improving gene editing in the liver, and dramatically improving T cell engineering, all potentially with lower toxicity.
Exactly. And why this knowledge is so valuable is that this synthetic method, plus the detailed understanding of why it works, that enhanced escape, gives us a powerful template to generate the next generation of ILs. It paves the way for more efficacious LNP-mediated delivery of biotherapeutics. Think protein replacement, better vaccines, more precise gene editing. It really broadens the horizon.
It really does. Which leaves us with a final thought. If these tiny, carefully designed structural tweaks at the ends of lipid molecules can unlock such powerful new delivery methods, what other hidden keys are just waiting to be found through molecular design? And how might discovering them completely change how we treat complex diseases in the future?